This is video illustration 4. The slideshow presented here is a brief introduction to the entire observation, recording, analysis, and graphic representation process used in IBSA's structural analysis research tactics. The slideshow begins with a repeated presentation of the brief sample video of original rat recordings seen in video illustration 3. The remainder of the video slideshow illustrates how a behavioral taxonomy similar to that used by Ray and Brown may be used for continuous coding of observations and how that results in time-stamped recording that allows us to conduct a subsequent kinematic analysis similar to that also reported by Ray and Brown in their 1975 and 76 publications. We'll begin by observing an animal for some period of time. If we have not yet created a set of categories to formally describe the behaviors of this species, we must begin by making a set of observational notes that describe the interactions we observe between the animal and its environment. We'll use simple descriptions, like the animal moving from one part of the chamber to another, rearing up to sniff and explore the upper portions of the chamber, moving again. Note that when the hind legs stop moving, the animal may continue to explore the chamber by sniffing, moving its head around, or even shifting positions without actually changing locations. We may even want to include all such variations into a single category, such as generally exploring aspects of the chamber. If the animal has had prior training, we may even want to elicit some behavior. For example, here is a delivery of water with our sound recording turned off so that CyberRat can use the video source materials. And the animal licks and drinks, then moves around and explores the chamber some more. We turn our notes into a standardized, mutually exclusive but exhaustive set of categories, generally called a taxonomy. Here's the one that Ray and Brown used in their research on rats in operant chambers, and we'll use it to illustrate their kinematic analysis. From the previous illustration of making notes, the categories should be relatively obvious as to what they describe. These categories were used to create a continuous, time-stamped data recording file using the panel at the lower right of this screen. Each behavioral event was coded as it ended by entering the corresponding keypad number or clicking on the abbreviation of the category. And the data illustrated in the table at the upper right is the result. Note that both the beginning time, as derived from the prior ending time, and the current ending time is recorded along with the category describing the activity across that time period. We'll actually play back a few lines to illustrate how each entry succeeds the previous to create a continuous description of all behaviors throughout an observation period. We begin with the animal exploring at the rear left of the chamber. Next, we see the first move from the back to the front left of the chamber. This move is followed by a very brief exploration and then another brief move. Then the animal rears. It subsequently moves across the chamber again, and so forth across the remainder of the video and its corresponding record. We'll ignore time in the subsequent analysis to be illustrated, but it's an important analysis in its own right and adds critical information regarding behavioral durations and time budgets. Our focus here is exclusively on kinematic analysis, which reveals sequential rules and associated conditional probabilities, describing paths of behavioral change as the animal engages with its environment. We begin with the data summary table in the upper left corner so that we can visually follow the data entries that we place into this sequential analysis matrix. Notice the taxonomy is repeated here twice, once on the x-axis, thus labeling each row of the matrix, and also across the top, thus labeling each column of the matrix. Each row, column, cell represents a sequence from one behavior to the subsequent behavior. So let's begin with our data entries by entering the first sequence that occurs in our data collection panel. Notice it is an explore to move. So we put a frequency count of 1 in that particular sequential cell. Moving to the next sequence, we now look at move as the beginning behavior and explore as the subsequent behavior. Thus we enter a frequency count of 1 in the move explore cell. The next sequence is an explore to move that indexes our previous count of 1 and makes it now 2 of those occurrences. Likewise, we'll enter a 1 count for move to rear up. Then a 1 count from the rearing up to the subsequent move. Another count indexing the move to explore now to 2. And so on through the matrix, adding 1 for each new sequential occurrence that appeared in the data analysis. When we're finished, we'll have a series of totals for each row. Those appear on our far right of the matrix, here in blue. But it's not appropriate to use raw numbers or frequency counts, as various durations of observations make these not very meaningful for comparisons. So we turn them into probabilities. Notice here the probability of behavior is not equal across all different categories. This is what Catania has described as the behavioral hierarchy. Move is the highest on the probability list of unconditional probabilities. It occurs half of the time. Explore occurs a third of the time, leaving lick, drink, and rear up to account for only 8/100ths of the time. Notice also that the cells have become conditional probabilities. That is, the 4 frequency count for explore to move has been divided by the row total of 4, thus making a probability of that prior condition, explore, changing to move as the subsequent behavior as 100%. Move to explore has a conditional probability of 0.67, move to lick drink 0.17, move to rear up 0.17, lick drink to move 1, and rear up to 1. But a kinematic matrix like this is not visually easy to decipher. So we use a graphing technique called a kinematic flowchart. The flowchart begins with the highest behavior on the hierarchy, in our case move. And move, remember, occurred half of the time. The question now is, what are the conditional probabilities given that move is our prior condition? Well, we have already determined that move goes to explore 2/3 of the time. Lick, drink, and rear up then occur and account for the other third. Likewise, explore always turns into move, lick, drink always turns into move, and rear up always turns into move. And note that our arrows indicate not only path or sequential direction, but also the conditional probabilities by virtue of the width of the arrows. Thus we have a kinematic flowchart that visually shows us a graph of all of the sequential probability paths. So that's the kinematic flowchart. Thank you for watching.